I'm Taki Shinohara, Deputy Director of Biostatistics and Professor of Biostatistics at Penn Medicine. To tell you a little bit about my education and background, I did my undergraduate and master's training at McGill University, where I trained in mathematics and statistics. I eventually moved into biostatistics and then entered a PhD program at Johns Hopkins, where I focused on causal inference and survival analysis, particularly multiple sclerosis research, and since then, that's been a primary focus. I'm a biostatistician who specializes in image analysis. Roughly a third of my time is spent in multiple sclerosis and neuroinflammation, another third in neuropsychiatry and neurodevelopment, and the rest of my time goes to collaborations across neurology and psychiatry more broadly. Lately, I've been spending more and more time on aging and dementia, and some work in epilepsy as well. The statistical methods we develop help translate biomedical imaging data into improved human health by enabling new therapies, improving on how we use current therapies, and creating more accurate and early diagnostic methods. To explain why this work matters, I'll highlight two main directions. First, in the diagnosis of multiple sclerosis. MS is an immune disease of the central nervous system that affects about 1 million Americans. While diagnosis is straightforward in some cases, other cases are complex. At a place like Penn, about 1 in 5 people who seek care for an MS diagnosis they received elsewhere don't actually have MS, they have some other condition. And many of these people receive powerful medications that they don't need, which can cause harmful side effects. The field is increasingly focused on more specific imaging biomarkers like the central vein sign and paramagnetic rim lesions, which were incorporated into the diagnostic criteria last October. Interpreting these advanced MRI sequences that reveal these biomarkers is challenging and time-consuming, and experts sometimes disagree on what they see. Our group has developed some of the first and most widely used automated tools that analyze those MRIs to determine whether MS-like biomarkers are present. This work should facilitate earlier, more accurate diagnosis and reduce misdiagnosis. Second, we work on multi-scanner image harmonization. Science is increasingly collaborative, and many studies pool imaging data from different sites and scanners. The problem is that if you take the same person and scan them on 2 different scanners, even from the same manufacturer, that can yield different quantitative results. Those inter-scanner biases can sometimes overwhelm the biological signals we're trying to detect. Our group has developed statistical tools and adapted methods to enable integration of data across scanners, so studies using multi-site imaging are more rigorous and powerful. That matters for everything from clinical trials of new drugs to large studies of healthy brain aging. Looking ahead, I'm excited to grow in several directions, especially in aging and dementia, and to move more of our tools into clinical practice. Many of the methods we've developed are currently implemented for research, but our goal is to make advanced imaging sequences and quantitative biomarkers more available in clinical settings, especially outside academic centers. We want to train and equip clinicians in the community to use these methods for patients who come through their doors. We'd like to focus on that translation into real-world clinical use going forward.